In the second recording of Chapter 16, we will focus on positive control and catabolite repression, TRP operon attenuation, and some additional level of gene expression control in bacteria. So, what is catabolite repression? We know that E. coli and many other bacteria preferentially metabolize glucose. Also, they can metabolize lactose and other complex sugar to yield glucose. If glucose is present, it would be metabolized preferentially, and it does make sense because glucose can enter glycolysis directly. It does not need any processing. So it would cost less energy to process glucose. And when glucose is available, therefore, genes that participate in the metabolism of other sugars are repressed. And we refer to this phenomenon as a catabolite repression. So, efficient transcription of lactoperon, for example, takes place only if lactose is present, however, only if glucose is absent. If there is glucose and lactose, lactoperon would not be turned on properly, and it is due to catabolite repression. So how repression can function as positive control mechanisms? This does not make sense, does it? It does actually. And this positive effect is actually exerted through the catabolite activator protein, CAP. So what does CAP do? CAP binds to the promoter of many operons, including lactoperon. So let's focus on lactoperon. We know that lactoperon is turned on if lactose is present, however, only if CAP is bound to promoter. And it is because RNA polymerase can only bind efficiently to promoter of lactoperon and many other operons only if there is CAP present bound to DNA in the promoter region. Now, how does this relate to the glucose and catabolite repression? So, CAP can bind to the promoter only if it's coupled to cyclic AMP. Only cyclic AMP CAP complex can bind to promoter. Now, cyclic AMP levels are inversely correlated to glucose level. So if glucose is high, cyclic AMP is low, and there is no formation of CAP-CMP, so there is no CAP binding to promoters, not to lac promoters. However, if glucose levels are low, cyclic AMP is high and CAP can bind with cyclic AMP, and at this point it can interact with DNA at promoter region, and it can enable efficient binding of RNA polymerase and exert a positive control on operons, including positive control of lactoperon. So here is just summary. This slide summarizes what we have discussed previously. So firstly, when glucose is low, so levels of cyclic AMP are high, and cyclic AMP can bind to CAP, catabolite activator protein, which now complex of CMP and CAP can bind to promoter and enable efficient binding of RNA polymerase. And if at the same time there was lactose present, so lactoperon will be turned on and transcription take place. So the lactoperon will be expressed. So CAP function as the positive control of lactoperon. However, when glucose is high, there is catabolite repression, because when glucose is high, cyclic LAMP levels are low, so there is no CAP-CMP binding, and in CAP alone cannot bind to the promoter. So RNA polymerase does not bind efficiently. So keep in mind, even if there is lactose and operator is free, so basically lock operon can be turned on. However, if RNA polymerase cannot efficiently bind to promoter and cannot start transcription efficiently, the efficient transcription will not be taking place. So catabolite repression refers to situation where glucose is high, cyclic AMP is low, so CAP cannot bind to promoter. Positive control, when glucose is low, CMP is high, CMP CAP coupled and now bind to promoter and enable efficient binding of RNA polymerase. The TRP operon of E. coli controls biosynthesis of the amino acid tryptophan. It is an example of negative repressible operon. So you recall, negative means that the regulatory protein is repressor and it must be synthesized in an inactive stage, so it cannot bind to operator, so operon is on, but it can be repressed. So TRP operon has 5 structural genes. The closer to 5' N is TRPE gene, following by TRPD, C, B, and A. This gene encodes 5 enzymes which together convert chorismate to tryptophan. So how does TRP operon works? Before we go to the regulation of TRP operon, I want to bring your attention to the 5' UTR, 5' untranslated region, which is very long in the TRP operon. And basically this 5' UTR plays a role in second level of regulation of tryptophan synthesis via attenuation. We will discuss attenuation in next slide. So let's go back to TRP operon. So what happens if the tryptophan levels here are low? The TRP operon is on. You recall the repressor is inactive, is synthesized in inactive form, so it cannot bind to operator. So RNA polymerase can proceed and can transcribe gene. TRPE is the first one, and every one of these gene encodes the enzyme which is involved in step-by-step conversion of chorismate into tryptophan. Now, when tryptophan accumulates in bacterial cells, so when tryptophan levels are high, tryptophan will bind to the repressor and it will change its conformation. So now the repressor can bind to operator and it can repress transcription of TRP operon. So tryptophan here function as co-repressor, and it's an example of feedback negative regulation. So let's now discuss attenuation. What is attenuation? Attenuation does not affect initiation of transcription. It does cause premature termination of transcription before transcription reaches structural genes by forming attenuator. Now, attenuation is one of the normal mechanisms of regulation of gene expression in bacteria besides repression. However, most of the operons are regulated either by repression or by attenuation. The only exemption from that is TRP operon. TRP operon is regulated by both, by repression, as we discussed in previous slides, and by attenuation. So let's go to next slide and see how attenuation works, what are attenuators and what are anti-terminators. So let's now discuss the attenuation. What is attenuation, how does it work, and why TRP synthesis, TRP operon is controlled by repression and attenuation. So as we mentioned before, the long 5' UTR plays a role in the attenuation. This long UTR is composed of 4 regions. Region 1 is complementary to region 2. Sequence of region 1 is complementary to region 2. Sequence of region 2 is complementary to region 3, and region 3 is complementary to region 4. So what does it mean? That region 1 and 2 can form a hairpins, and regions 3 and 4 can form a hairpins. After region 4, there is a string of the uracil. This string of uracil follows the 3 to 4 hairpins. If there is hairpins followed by string of uracil, the attenuation take place. This structure is called attenuator because if this structure forms, the transcription of mRNA will be attenuated, so there would be no further transcription. However, if there is formation of complementary loop between 2 and 3 right here, so there is no hairpins followed by poly U, so in this case the structure does not terminate transcription and transcription proceed. So first scenario, when we have attenuator formed, happens when tryptophan levels are high. The second scenario right here, when there is no attenuation, so this is for this loop 2 and 3 is called anti-terminator, happen when tryptophan levels are low. So how this is controlled? Important part in this play translation. So keep in mind that in bacteria, when transcription starts, translation proceeds also. So at the same time you have transcription and translation of the mRNA. Now in region 1 right here, there are two UGG codon encoding tryptophan. And this tryptophan codon regulate whether there will be attenuator or anti-terminator formed. How? Let's go to next slide. So how attenuation works? The best explanation you can find is in the animation you have in Launchpad. I strongly advise you go back and watch that animation. It explains attenuation in great detail and very clearly. I will try to outline only the major point here in attenuation. So let's first start with the condition or under the circumstances when tryptophan level is low. Remember, transcription start at 5' and 5' UTR has these 4 regions, and in region 1 there are 2 TRP codons. So as transcription starts, mRNA is synthesized. It proceeds through region 1, region 2, and so on, so on. You remember that in bacteria, while transcription is going on, translation start. So as transcription is going on, the ribosomes attaches and start to synthesize a protein, on the region 1. Now, there is one thing I want to bring your attention at. UTRs are untranslated regions, so UTRs normally do not encode proteins. However, TRP operon is an exception again. 5' UTR of TRP operon encodes short protein, which is however unstable. Seems like this protein its only function is to regulate attenuation. So even though the 5' UTR encodes short proteins, this protein is unstable and does not play a role in structure or enzymatic metabolism of bacteria. So let's come back here. So once the ribosome reaches those TRP codons in region 1, it stalls, because if tryptophan is low, there are very few tRNA loaded with tryptophan. So basically the ribosome stalls and waits for tRNA which carries tryptophan. However, transcription continues. So region 1 is blocked by ribosome, so it cannot form the loop with region 2, but transcription continues. So region 2 and 3 are transcribed while translation is stalled. So in this case, basically the region 2 and 3 would form the loop, which is anti-terminator, because it will not trigger attenuation. The ribosome eventually received tRNA carrying tryptophan, so it start to move and continue translation, and it can overcome this 2-3 loop. So this 2-3 loop does not terminate transcription. However, when tryptophan levels are high, right here in second part of this figure, in this case, so the transcription starts followed by translation. Since tryptophan levels are high, there is high level of tRNAs which are loaded with tryptophan. So when ribosomes arrive at region 1, it can easily proceed. So translation closely follows transcription, so it releases region 1 and 2. So basically region 1 and 2 can form the hairpin secondary loop, and therefore after region 3 and 4 are transcribed, they form attenuator. You remember region 3 and 4 are followed by the string of uracil. So hairpin followed by string of uracil triggers release of RNA, mRNA from template, and actually it triggers termination, attenuation, termination of the transcription. So why does TRP operon needs dual regulation, and what is the difference? If you recall, repression of TRP operon is controlled by level of tryptophan. Attenuation is controlled by availability of tRNAs carrying tryptophan. So why is the second mechanism of control of TRP operon expression? The best guess is, or what was suggested is, similarly to lag operon, TRP operon is never shut down completely, is never suppressed completely. So attenuation increases repression of the TRP operon to higher level. So it does contribute to efficacy of the suppression of TRP operon. So besides the operons and attenuation, expression of bacterial genes can be controlled at the additional levels through the antisense RNAs, riboswitches, or ribozymes. Certain genes in bacteria are controlled by the antisense RNA. Antisense RNAs are short RNA which are complementary to particular sequence in mRNA. You have an example here of OMPF gene. This gene, outlined right here, encodes a protein which functions as a channel for the passive diffusion of small polar molecules such as water or ions. Under normal condition, when extracellular osmolarity is low, the OMPF gene is transcribed and translated and forms this protein, the channel, and allows the transport of diffusion of these molecules. However, when extracellular osmolarity increases, this increase in osmolarity triggers transcription of MICF gene. This MICF gene encodes RNA which is antisense, which is complementary to mRNA of OMPF gene. So when this RNA is transcribed, it does anneal to OMPF gene mRNA and inhibits its translation. So it blocks ribosomal binding site on OMPF mRNA, so there is no protein translation, protein synthesis. Now, be aware that antisense RNA in bacteria are not siRNAs. They are not processed by dicer. They do not associate with RISC complex. So also they are complementary to mRNA and inhibit translation. They are antisense. They are not siRNAs. Riboswitches represent additional level of gene expression control in bacteria through the mRNA. Riboswitches are regions usually found in 5' UTR of mRNA, which form secondary structures. The secondary structure can take different form depending whether they interact with regulatory proteins or not. This regulatory protein can either enhance or trigger translation of mRNA, or it can inhibit translation of mRNA. So both scenario can happen depending on which riboswitch in which gene in which mRNA is interacting with regulatory protein. Under scenario outlined here, you see here a riboswitch, which is complex secondary structure. If riboswitch interacts with regulatory protein under this scenario, this riboswitch is stabilized in secondary structure that masks ribosome binding site, and therefore no translation takes place. In the same riboswitch, if the regulatory protein is released, riboswitch secondary structure open, it changes conformation, and now ribosome binding site is open, and it can now interact with ribosome and initiate translation of the protein. One of the examples of the gene regulated by riboswitches is synthesis of vitamin B12 by bacteria. So when a vitamin B12 is found in an activated form. In this activated form, riboswitch falls into secondary structure that actually masks the ribosome binding site, and there is no translation, no B12 synthesis. However, if there is no activated form of vitamin B12, so riboswitch has conformation which enables binding of ribosomes and translation and synthesis of vitamin B12. So last point I would like to make is that riboswitches can function as ribozymes. You know, the RNA molecule, some RNA molecule have enzymatic activity, and we refer to them as ribozymes. So here is an outline of the riboswitch, which upon binding with regulatory protein not only changes conformation, but it also activates its ribozymes function, and it self-cleaves the mRNA and therefore degrades mRNA and terminates the translation.